Hello everyone. So today we are going to understand about the topic electron transport chain. But first let us go through a little bit that we have been studying in the last two videos, that is aerobic respiration. So in aerobic respiration there are total four steps. Firstly, there is glycolysis, where we will see that not only ATP, NADH is also released, and then we can see in the link reaction when the pyruvic acid that is formed, it is entering into the mitochondrial matrix. It undergoes link reaction and then finally it enters into the Krebs cycle. So in link reaction and Krebs cycle, we will also see NADH is also released. But you all might be wondering that what we will do with this NADH, right? Now is the time we will get to know in the electron transport chain how we are going to get the ATP from our NADH. So we can consider that NADH and FADH that has formed here, these are basically our like indirect ATP, we can tell, because by oxidizing them, we are going to get ATP. So that's why we can easily say they are indirect ATP. Now, for getting this indirect ATP, we need to understand the electron transport chain. But this electron transport chain which occurs here, basically oxidative phosphorylation takes place. That means, of course, there is going to be oxidation and phosphorylation. Why oxidation? Because the NADH and FADH, when they will be oxidized, they are going to release H plus. Now, this H plus are going to be pumped into the intermembrane space. So the intermembrane space is basically, we know that the mitochondria is a double membrane structure, right? So what we will notice that this portion that is present between these two membrane, this is the intermembrane space. Okay, so the intermembrane space will now have high concentration of H plus. We will increase the H plus concentration. And how we are going to increase? We are increasing by using certain complex which are going to pump the H plus from the matrix to the intermembrane space. Now, when there will be increase in H plus in the intermembrane space, so we can write high H plus concentration, and the matrix will have low H plus concentration. So you all can see there is a proton gradient or H plus gradient that has been created. Now, this is going to help us in production of ATP. So obviously phosphorylation will take place. So let us first understand about the electron transport chain. So not only ATP will be forming, we will see that electrons are going to be also transported by when we are going to see oxidation takes place. So that time electrons are going to be released as well. So let us first understand about electron transport chain. So you can see there are total four complexes which are involved. Okay, so the electron passes from one carrier to another via this complex 1 and 4 in the electron transport chain, and there will be also complex 5, that is ATP synthesis, which is helping in pumping the the H plus which has been transported to the intermembrane space again back to the matrix. But first, let's understand about complexes. So complex 1, that is NAD dehydrogenase, it is going to oxidize our NADH, and as NADH is going to be oxidized, it is going to release 2 electron. It is going to be FMN, that is flavin mononucleotide, and it is going to the iron sulfur cluster. So this is FES, is iron sulfur cluster, and finally it is going to the ubiquinone. That means the ubiquinone that you can see here, it is a mobile electron carrier. So from complex 1, the electron is also going to come to ubiquinone, and we will see the complex 2, that is succinate dehydrogenase. From here also, the electron is going to come when the FADH is going to be oxidized. So from complex 1 and complex 2, electrons are coming to this ubiquinone. Now this ubiquinone, it is going to be reduced. So the we will see that it is forming reduced ubiquinone, which is also called as ubiquinol. Okay, so this is a electron carrier. So please remember this UQ is basically ubiquinone electron carrier. So the two electrons which are coming here, then they are going to our complex 3 and then finally complex 4. But Before going to complex 4, we will see there is another electron carrier, that is cytochrome C also involved. Okay, this is simple one I have drawn, but it is not so simple, but I am going to make it simpler for all of you. So you can see from ubiquinone, the electrons are going to cytochrome B, then iron sulfur cluster, and to C1, as the name is also cytochrome BC1 complex. Okay, this is complex 3, and then we will see again another mobile electron carrier, cytochrome C is responsible, which we can see it is in the outermost part of the like inner mitochondrial membrane, and it is going to transport the electron to our complex 4. But the name of the complex 4, you can see it is cytochrome oxidase, because cytochrome C is being oxidized due to this electron transport, right? And we will see it as, that's why the name is cytochrome C oxidase. And in cytochrome C oxidase, there are two copper atoms. You can see CuA, and this is CuB. Along with that, that is site A and site A3 as well. Okay. Now, where will this electron go? Will it go to the complex 5? No, it is not going to complex 5. Rather, the electrons are going to be now transported, or who is going to finally accept this electron? So the final acceptor of electron in electron transport chain is our oxygen. So finally we can see the role of oxygen in the aerobic respiration finally. Okay, so oxygen is going to accept it, and it is also going to take the help of two H plus, that is proton present in the matrix, and this two electron, and form H2O, that is water. So water is also forming, we got to understand about it. But along with this, we are going to see that the protons are pumped also by these complexes. Complex 1 is going to pump proton, complex 2 is not going to pump any proton, complex 3 is going to pump proton, and complex 4 as well. But complex 4 is only pumping 2 proton, because 2 protons are being utilized during formation of the water. Okay, but we can notice in the intermembrane space now there is high H plus concentration, isn't it? We can see there is high H plus concentration. Now, if you notice, the high H plus concentration is now present. So in the matrix, what we will see? In the matrix, there will be low H plus concentration. Of course, there is going to be low H plus concentration. So what will the proton want? They will again want to come back to the that is our matrix from the intermembrane space, but they cannot, because this inner mitochondrial membrane, the inner membrane of mitochondria, is impermeable to H plus, okay, ions. Okay, so now what is going to happen? Our complex 5 is our savior. So how is complex 5 going to save us? So you can see in complex 5, we have a headpiece, which is basically the peripheral membrane protein complex that is containing the site of synthesis. So you can see the ATP is synthesized, and we have the F0 membrane, which is a channel through which the protons are going to come back. So the H plus ions which are present in the inner membrane space, they have to come back to the matrix, and for this they are going to take the help of the complex 5, that is ATP synthase, which is containing two subunits, that is F0 and F1. F0 is the channel, which is a F0 is a integral membrane protein, and F1 is a peripheral membrane protein. So the passage of proton through this channel is coupled by catalytic site of the F1 component. So in the F1 component, there is a catalytic component which is helping in production of the ATP, and we will see that when 4H plus 4 protons are going to be passing through this complex 5, it will release 1 ATP. That means 1H plus is going to give us 1 by 4 ATP. Right, class? We have understood about it. Now, how is this happening, right? There is a theory behind it that is called as chemiosmotic theory. So you can see there is a proton gradient, or rather we can see it is a electrochemical gradient, okay? So as there is increase of the H plus, so pH also increases, and electrical gradient is also going to form. So a we can say electrochemical gradient is forming. How it is forming? Because of the electron flow which is accompanied by proton transfer. So not only we see that there is a electron flow, proton transfer, both are taking place. So the like the chemical gradient and the electrical gradient, they are going to help in the ATP synthesis, which is driven by the proton motive force. So why will the proton come? As I was telling, that inner membrane space, this portion is the inner membrane space. This is going to be impermeable to our protons. They cannot come, so they are going to take the help of our complex 5 for this, right? So the inner mitochondrial membrane is impermeable. Now they take the help of the complex 5 to re-enter into the matrix, and this is possible because the proton motive force that drives the proton back into the matrix, and this is why we can see there is production of ATP, because it provides the energy. So as the proton motive force is there, so the energy is entering, like is provided, and that is how the ATP is synthesized. Okay, and for ATP synthesis we have seen that F0 and F1 complex is helping in production as well. So F0 is transporting the like the protons, and F1 having the catalytic side, it is helping in production of ATP. Okay, and this chemiosmotic theory was proposed by Peter Michel. All right, so I hope all of you have understood about it. Okay, so now we are going to focus on our balance sheet of our aerobic respiration. But before we understand about the like the balance sheet, we have to understand few features of this, like how many ATPs are released. For that, I am going to use new slide. So if you remember, students, I have been telling all of you that 4H plus is going to provide us 1 ATP. That means 1 H plus is going to give us, so we can write 1 H plus is equal to 1 by 4 ATP. Now, we know that when one NADH is there, right? For NADH, what we will see? Let's draw complexes. Okay, complex 1, complex 2, complex 3, and we have complex 4. So the complex 2 is not going to work for NADH, right? Because it is not going to transport the electron after oxidation of the NADH. It is responsible for the FADH oxidation. So we will see NADH, it is going to pump 4H plus, and the third complex is going to again transport 4 electron, and the fourth one, it is going to transport the 2 electron, 2 proton. So finally we are getting how many? 10 proton, right, from 1 NADH. So that means 1 NADH, it is going to give 10 H plus. That means how many ATP? That means 10 into 1 by 4. It is going to give us 2.5 ATP. But students, when we are going to do the calculation, are we really going to consider 2.5 ATP? No, we are not going to consider. Please remember this. We do not consider this in case of your NCERT books. But when you go to college or in your like higher education, you will see it is 2.5 ATP. But when we will calculating the number of ATP that we are getting from the NADH, we are going to consider 3 ATP. Please remember this. The calculation is not there in your book, but just to give you an idea, I have explained about it. Now let's come to FADH. When FADH is going to be released and how many proton it is going to give. So let's come to complex 1, complex 2, and we have the complex 3 and complex 4, right? So complex 1, it is not going to work because it is NAD dehydrogenase. Complex 2, it works, but it is not going to give any kind of proton, right? We will see complex 4, it is going to give 4 proton, and we have complex 2, which is giving 2 proton. So ultimately we can see 6 protons are going to be released. So 6 protons, what does it mean? 1 FADH, it is going to give us 6 into 1 by 4, it is going to give us 1.5 ATP. But again, I am telling you, we do not consider this. We consider 1 FADH is going to give 2 ATP when you are studying for your NCERT or your NEET. Okay, you will be studying NADH 3 ATP and we have our FADH 2 ATP. Okay. Now, before we come to our calculation about the balance sheet, before understanding, you have to understand when we are calculating this respiration balance sheet, we are considering certain assumption. Okay, it is not actually happening in our cell. In our cell, we will see not only the glucose will be oxidized and it is giving us energy when we are starving. Okay, that time we will see also fat is also getting broken down and it is forming into fatty acid. It is performing certain like the steps and certain reaction and it is producing acetyl CoA, or it might be also entering through that is dihydroxy acetone phosphate as well. So this can happen. Okay, I'm not telling this cannot happen, but the main thing is that we won't be considering that fat or the protein. Even protein is also a respiratory substrate, okay, which enters into the respiration. But we are not going to consider this as a respiratory substrate. So the assumption first we are going to make that there are sequential pathway where we will see, and the NADH which was produced in the glycolysis. So if you all remember, I have been teaching you this glycolysis, right? So in glycolysis we have seen there was not only ATP, the NADH was also released. Then we have also studied that in the link reaction also the NADH was released, and then we have also studied that in the citric acid cycle NADH as well as FADH, all this were released. So we are going to all consider this, okay, when we are going to calculate. And then none of the intermediates, so there might be certain intermediates which are going to be produced while this glucose 6 phosphate or fructose 6 phosphates are produced. There might be certain intermediates which are formed. So none of the intermediates in the pathway are utilized to synthesize any other compounds. They won't be producing any other compounds, we will consider, but actually it doesn't happen in cell class. Now only glucose is respired. As I was telling you, we are not going to consider fat or the protein. Only glucose is going to be respired, and it is going to give us all this like ATPs and also NADH and FADH. Okay, so it is quite simple now. So let us now come. What will happen when one glucose molecule is going to be oxidized? So here is the balance sheet. So the first glycolysis we have seen, there were various steps, total 10 steps where we have studied, right? So out of this 10 steps, the first step we will see that ATP is utilized. So we are going to write -1. Again in the third step, fructose 6 phosphate to fructose 1,6 bis phosphate, we will see again 1 ATP is utilized. So 2 ATP already we have seen -2 has happened. So now glycerol 3 phosphate 1,3 bisphosphoglycerate. Now if you all see from here onwards, I have written in every molecule it is 2. Why so? Because if we come to our glycolysis, if you see students that in the payoff phase, all the molecules that has formed, they are forming twice, because the triose phosphate, that is a PGAL which has formed, it is a 3-carbon compound, and that is why twice the reaction is going to be happening. Okay, so all this which is happening, it is twice, because 2 pyruvic acid, we will see 2 acetyl CoA, right? So now let us focus on the balance sheet. So we will see here that the glycerol 3 phosphate that was forming into 1,3 bisphosphoglycerate by the dehydrogenation. We will see 2 NADH was released, and as I told you when I was calculating this, that we do not consider 2.5, rather we are considering 3 ATP for 1 NADH. So please multiply 3 whenever you are calculating NADH. Okay, 2 into 3 ATP, 6 ATP. Then again, we have seen that ATP was released, 2 ATP released in 1,3 bisphosphoglycerate to 3-phosphoglycerate, that is the 7th step. And then we see the 10th step is also where the ATP is released. So 2 ATP is released. So finally we can see up till here 6 plus 2 plus 10, 16. Okay, sorry. So my bad. So it is going to be 6 plus 2, so 10, 10 minus 2, it is going to be 8, right? I hope it is clear. This much it is coming 10, but what we are doing 10, and here we have -2. So 10 minus 2, it is coming as 8. So in glycolysis, we are getting total 8 ATP. Okay. Now the link reaction, as you see, the pyruvate, the pyruvic acid is forming acetyl CoA. Again, there is going to be a oxidative decarboxylation where NADH is going to be produced. So 3 multiply we are going to do. So 6 ATP is produced in the link reaction. Then finally, in the 8 steps of the Krebs cycle, we can see in the first step, there is no ATP or any kind of NADH release. But in the third step, we can see isocitrate is forming alpha ketoglutarate acid because of oxidative decarboxylation. 2 NADH is produced, so 6 ATP. Again, in alpha ketoglutarate, 2 succinyl CoA, 2 NADH is going to be released, so 6 ATP again. And succinyl CoA to succinic acid, we have seen there was a substitutable phosphorylation, so 2 ATP or GTP is released. So it is 2 ATP we have written. And succinic acid to fumaric acid, again there was a dehydrogenation, but here not the NADH but FADH is going to be released. So we are multiplying by 2, okay, because we have studied 1 FADH is 2 ATP. We are considering this. Now if we, okay, sorry, we are coming to this. Now we will see 4 ATP are going to form. Now malic acid to oxalic acetic acid. So again 2 NADH, so it is going to be 6 ATP. So this comes as 24. So in 24 ATPs are going to be released from the oxidation of the acetyl CoA, right? So finally from 1 glucose we can say 38 ATP, but it is not the case that always there is going to be 38 ATP. In a certain case, we will say that there might be 36 ATP, and it happens when. So as I was telling you that the pyruvic acid, it is going to be transported into the mitochondrial matrix. So that time the shuttles that are going to help, they might charge you some money. So they are going to charge you some ATP, but it is not for all the shuttles. The malate aspartate shuttle that we see in the liver, kidney and the heart, they are not going to charge. They are free of cost, though they are not going to take any ATP. So they are going to give 38 ATP only in this cell. But in the skeletal, muscle and the brain cell, we are going to see that there is going to be 36 ATP because 2 ATPs are released in case of the glycerol 3-phosphate shuttle. Okay, so that is how we have understood that how it is forming. So let me just give you like a recap. So if we say complex 1, in complex 1 we will see NADH, right? And NAD, it is oxidized by NAD dehydrogenase. 2 electrons are going to participate. So what we see FMN, and it is going to FES, iron sulfur cluster. It is going to ubiquinone, UQ. And we can see the complex 2 is also here. So let me draw it with another color. So in complex 2, we will see that succinate dehydrogenase is working. So we will see that FAD is going to be FADH is going to be oxidized, right? So the FADH is going to be, okay, I'm writing NADH, sorry, my bad. So FADH, which has been oxidized here, so we will see FAD, it is going to transfer 2 electron to FES again, and it is going to come to the ubiquinone. So ubiquinone, it is going to reduce into UQH2, and we will see after this there is going to be transfer of the electron to the complex 3. The complex 3 is basically cytochrome B C1 complex. So we will see cytochrome B to FES to cytochrome C1, and then it is going to cytochrome. Okay, let me take another color. Okay, so it is going to be cytochrome C mobile carriers. So these are all the mobile carriers that we can see. Now from here we are going to see it is going to the complex 4. So in the complex 4, what we have noticed that in complex 4 there is going to be cytochrome C oxidase, because the cytochrome is oxidized here. As the cytochrome is being oxidized here, we are going to consider that. So here we will see 2 copper atoms are there, CuA, then it will go to site A1. Okay, so there are 2 sites which are there, then it is going to CuB, and then finally site A3. And after this, it is going to go into the oxygen. So we will see that the half O2 is going to accept the electron. So the two electrons are going to come. So the half electrons is there, and we will see along with this there is going to be two H plus, which are going to give us the H2O, right? Water. But where is the H plus being pumped? So we will see. Let's consider now. Let us draw. So this is our complex 1, and this is our complex 2, and this is our complex 3, and this is our complex 4. Okay. So I hope now it is little bit easier how to do it. And who are going to pump? So we will see that this is going to pump 4H plus, right? And we will see this is also going to pump 4H plus, but only the four that is our site C oxidase, it is not going to pump. Okay. So if I make this little bit smaller, okay. So I have made it a little bit smaller for all of you. So we can draw it very well. Okay. So now what to do? Let me erase it here and draw it well. So we will see which are the that is complexes which are going to give us the proton. So we will see the complex 1, it is going to give 4H plus, complex 3 is going to give us 4H plus, and complex 2, it is going to give us 2H plus. Okay. So I hope now it is clear for all of you. Okay, you can all practice with the help of this diagram. And where from the proton is coming again? We will see from the IMAs, the protons are again coming back to the matrix. And for that, we require our ATP synthase. Okay, remember this. And along with that, we are also getting the ATP as well. Okay, I hope up till this it is clear, all of you. I hope now all of you can see it is pumping the H plus to us. Along with that, we will see it is also going to give us the ATP as well. Okay. So all these, like we can see the complexes which are present, they are going to be present where? They are going to be present in the inner mitochondrial membrane only. Okay, please remember this. All these complexes are here, and we are increasing the gradient in the intermembrane space. And we have seen as there is a proton gradient, the ATP is going to produced by the proton motive force. We will also see it is also like taking all this H plus which are present in the intermembrane space into the matrix as well. Okay. So I hope this calculation is also clear, and how aerobic respiration happens and how the shuttles are going to take some ATP as well. The students, make sure all of you are practicing. Okay, practice is the key to success, and have a nice day everyone. Take care. Bye bye.